Hello everyone. Welcome to the first video lecture on Introduction to Immunology, a fascinating branch of biology that explores how our body fights with the diseases. My name is Dr. Vipin Verma, and today we are diving into the that forms the foundation of this complex yet vital field. Whether it is a common cold or vaccine or any autoimmune disease, immunology is at the heart of our understanding, treatment, and prevention. I suggest not to skip any content or not to fast forward, as you may miss some crucial information which you may not connect with the future content. My email is also given in case if you wish to share your feedback. So let us start this exciting journey. So please give your attention to the screen. If you see the immunity is the state of protection which is inside our body, and the term immunity itself comes from the Latin term which is uh, immunis or immunitas, and that means freedom from. Now, this term is very old. It it if you trace it back, you will find that the term was taken in English from the Roman Empire. In Rome, like we have the parliament, they used to have a senate. Like we have the member of parliaments or MPs in Lok Sabha and Rajya Sabha, they used to have the senators. And these senators were very close to the king, and they were very influential people. They were like the commanders or the wealthy businessmen or the royals who were the part of the senate, and all the senators, they were exempted from any legal trial. That means they were immune, or they had the king's immunity. Nobody could try them in the court, whether they do murder or whether they do any kind of crime. Nobody had the right to try them inside the court. And that's why this term was taken, and this term came into English, and it was finally became the part of our immune system, because our immune system or immunity is the state of the protection. Now, it is interesting to know that when the research on immunology started, it started with the quest to understand how our body reacts or fights against pathogens, or those agents that give infection to the body. We call them as pathogens. But later, when the field expanded and when the research expanded, it came to us, and it was later found out we that the immunity is not only protecting us from the pathogens, but also from a number of other environmental agents. And here you can see these can include, apart from the microbes, these can include the food. You must have heard that many people have food allergies. They eat something and immediately their body starts showing certain reactions. So that food allergy is because the body is not tolerating the food. Then certain chemicals. Some people, they may have allergy to the nail paints or the wall paints or any other normal chemical, even the detergents. People have eczema, so that is a kind of chemical allergy. Then pollens. I mean, this is very common that you will find the seasonal allergy due to the pollens, and people sneeze. They develop the hay fever. It is all because of the pollens which are spreaded in the environment in certain seasons, particularly the spring seasons. Then drug allergies. Some people are very allergic to certain drugs. One prominent politician, she was very allergic to this the band-aid. you know and then some internal components, for example cancer and tumors, because cancer and tumors are the part of our own body. The cells are our own body cells, but when they show mutations and they start expressing certain molecules which are not the part of our own body system, then our immune system attacks those cells and eliminate them. So you can see that immunity is just not only giving us protection against the microbes or the pathogens, but they are also giving us protection against a number of environmental agents. So now, as I said, that it was evolved to fight pathogens invaded in our body. And when we say pathogens, we have the four categories of pathogens. The first is the bacteria. So I am giving the example of streptococci. Now you can see here these cocci. Cocci is the plural form of coccus. So coccus means round shape. So here you can see so many round shaped bacteria, and they are in the form of a chain. So they are called streptococci. So streptococci is a bacteria which is responsible to give you the strep throat, the throat infection. You have a bad throat, and then you have some cough and you have discomfort. So that comes of this particular bacteria, which is the streptococci, right? So this is an example of the bacteria. The next example is a virus. Now this is the picture of a rabies virus, and you all know that rabies is 100% fatal disease. So that means once the virus goes inside the central nervous system, then it is really almost impossible to escape death. So rabies is a kind of virus, streptococci is a kind of bacteria. The third category of pathogen is a mold. Now, mold is a example of a fungus. Now, fungus is it is neither animal nor a plant, but it is a different kingdom, separate kingdom. And I'm sure you must have seen this mold growing on the old breads. Or if you have not seen, then what you do, you can just take a bread piece, put some water, sprinkle some water on it, and leave it overnight, or maybe a 2 or 3 days, and you will find the beautiful growth of this fungus on your bread, right? So mold is an example of fungus. And the fourth example is the kind of worm. So here the example is Wuchereria bancrofti, which causes encephalitis, right? So these four categories of pathogen, the bacteria, virus, mold or fungus, and worms, they cause a number of different diseases in body. Okay. Now, if we see a lot of pathogenic diversity. If you see the polio virus, the size of the polio virus is around 30 nanometer. Now, 30 nanometer is extremely small, very very minute particle. On the other hand, we also have renal worm, which is around 100 centimeter. Now, 100 centimeter is almost 1 meter. So you can see the variation in the pathogens which infect our body. So renal worms, it can be a meter long, and if you see, it can be 100 centimeter by around 1 centimeter. So 1 centimeter is the width and 100 centimeter is the length. So our immune system, they have different strategy to fight different kind of microorganisms, because viruses can easily be taken up, and they are definitely, they need a cell to replicate themselves. So we have a different strategy. On the other hand, worms cannot be taken up by any cell. No cell can phagocytose the worms. So that means these such worms, they will be surrounded by a number of cells which will secrete a lot of toxins so that the worms can die. Okay, so we have a different strategy for different organisms, and that's the beauty of our immune system. Then if you see our immune system, it is an interconnected network. It is an interconnected network of cells, molecules, tissues, pathways, and organs. And these things, they work together to defend the body against infection and diseases. So sometimes it is so complex that it is, difficult to understand or difficult to start with. Where to start with? Whether we should start, to study from the molecules, or from the pathways, or from the cells, or from the tissues, or from the organs, or what is the perspective where we should start at. So it is so interconnected and so intertwined, but it gives a very robust, protection to our body, and that's why it keeps our body healthy. Then this immunology has led to the development of vaccines, and in fact, if you see the foundation of modern immunology, it actually started with the vaccines, which I will tell you. Then successful transplantation. If a person needs transplantation and it needs an organ, then definitely, and that organ is from, the different person, that we have to, you know understand the immunology of this transplantation to be successful. And then the drug development, because a lot of understanding have come to know, about the different diseases, and now we know how the diseases are caused. And because of the understanding of the immunology, we can easily, develop the drug. For example, we have developed the drug for asthma, because we know that asthma is because of some dust allergies or mite allergies or the pollen allergies, where our bronchi, they swell, they inflamed. And when we have this information, it is easy to develop the drug. So immunology is an important area where, it is giving us a lot of information to develop vaccines, successful transplantation, and successful drugs against a number of diseases. Now, if we see the immune components, these immune components are found throughout the living world. It is not that only humans have the immune system, but if you see the other animals or the plants, you will find that some other elements are always present there. Now we will give some example here, so that you will have some understanding about this. In plants, resins, they are produced by gymnosperms, and they provide defense against fungal and insect attacks. So here in the picture you can see, this resin droplet which is oozing out from the trunk of a gymnosperm. So this gives a defense against a fungal and the insect attacks, right? So it is a kind of immunity which gymnosperms have developed. Then some phytoalexins, they are the antimicrobial compounds which are accumulated by plants temporarily. In some stages, phytoalexins, these are the compounds which are accumulated, and they show the antimicrobial activity. Now, even in the lowest invertebrates, which are the sponges, you can find the picture of the sponges here. They have the cell surface proteins that mediate cell recognition in a very basic immune system. So, if you see, you must have heard about the different kingdoms, and if you see the animal taxonomy, you will find that Porifera is at the base of this taxonomic division. And even in porifera, where the sponges are the example, they have the cell surface proteins which mediate cell recognition, and that's a very basic immune system. Now if you go further, then we have the drosophila, and drosophila is an insect, right? Drosophila is an insect, and it's a part of the orthopoda. Now it produces some antifungal peptides, and whatever information is given here, this is all based on the research. So that means that the elements of the immune system are not only present in the humans or the higher vertebrates, but also they are present in the lower vertebrates as well as in plants. Now, as you go move up in the taxonomic ladder, on the taxonomic ladder, you will find that the immune system becomes more and more complicated, and it is becoming more and more complex, right? So humans have the most complex immune system in terms, if we assume this particular hypothesis, and which is true, right? So now, one term which comes is the herd immunity. Now, herd immunity is a concept where a population becomes protected from an infection when a large percentage of individuals are immune. That means immunity is a state of protection, but not all individuals have the same immunity, right? For example, if you see a class, in class there may be many students, and all students not have the same, they will never have the same level of immunity. Some students, if there is some cold or flu is going on, then in that case some students, they may have, very frequent infection, they may be sneezing, but some students, despite of surrounded by such microorganisms, they don't catch the infection. Even they catch the infection, but because they have the very healthy immune system or the stronger immune system than the other students, they are protected. So that means that in a small class, this is a scenario of a small class where suppose there are 30 students or 50 students. Now let us talk about a big population. Let us talk about the country. For example, if we talk about the city or a village or a state or a country, now it has a lot of population, a very high number of people they live, and when these people, some will catch infection, some are immune, some will catch infection, some are immune, and they are spread together. Now what happens, the concept of herd immunity is that a population becomes protected from an infection when a large percentage of individuals are immune. Now how it happens that it can be acquired through vaccination or recovering from the disease. Now, you must have seen this particular example during the recent COVID 19 outbreak, where after 2 cycles or 3 cycles, now the COVID is not that virulent. The first 2 cycles were very virulent and it caused a number of deaths. But slowly, because of this infection and the successful vaccination program by the government, the population, it started gaining immunity against COVID-19 virus. And now if you see that COVID-19, it comes seasonally, or sometimes you hear the cases here and there, but they are very sporadic cases, or very a very small number of cases. Why? Because the population has achieved this herd immunity. Most of the population is immune or against this particular virus. Why? Because of the natural infection. The people who did not die, who had the disease, they developed the immunity, or by the vaccination, who took the vaccines but who were prone, they developed the immunity, and that created a barrier that those who did not take the vaccine, or those who who are born after the COVID-19, or the individuals who were very susceptible or vulnerable, they did not, or they will not catch this disease, right? Or they will be protected to some extent. It is the concept of the herd immunity, and it can be acquired through vaccination or recovering from the disease. Now it works by the immunity threshold. How it works? How herd immunity works? By the immunity threshold, where the percentage of immune individuals, which varies based on the disease transmissibility, right? The percentage of immune individuals, which varies based on the disease transmissibility. If we have a higher number of individuals which are immune, our herd immunity will be very strong. If we have the smaller number of people who are immune to a particular disease, then our herd immunity is a bit weak. So we need to have an immunity threshold. Now threshold is the level we need to have the immunized people in a population where the disease can be protected, or can be prevented through this particular barrier, right? So that is the immunity threshold. Then herd immunity also works by the protection mechanism. When the people are vulnerable but they are surrounded by the strong immune people who already have the high immunity against a particular disease, then they have a reduced chance of exposure, because the person who has a immunity against a particular disease, he will not spread this infection further, because he will not have that symptom. By sneezing or by some other means, it will not spread that. And therefore the person who is vulnerable or who has not developed this particular disease, he will be protected. So vaccination is the most effective way to achieve herd immunity as compared to natural infection. Why? Because we cannot allow natural infection to go to the population, and then we can see whether our population is particularly immunized or not. Why? Because it will be very slow process, right? Or it may be very risky process, right? But vaccination, once we have developed the vaccine and once our population has taken the vaccine, then it is a very short effective way to create a barrier, to create this herd immunity threshold, to bring this up in a very quick time. So vaccination is the most effective way to achieve herd immunity as compared to the natural infection. Now in this diagram, if you see here, there is no herd immunity. So this particular individual is infected, and it is then spreading the infection in the society through its own near and dear ones, and then they are giving the infection to the distant ones. So you can see here that the number of infected people are more, and when we have achieved the herd immunity, so these green one, they are the immune people. If you can see, these all green one are the immune people. And this person, even if he has a disease and he is giving to the individual, but since these immune people have created a barrier in the society, then vulnerable people or susceptible people, they are protected, right? So the disease is not going beyond this person, because there is a barrier of a number of persons who have achieved the immunity. So that is the concept of the herd immunity, where we give protection to the people by vaccination, right? Or even if the vaccines are not available, then by the natural infection, after some time or after few cycles of infection, the population gets the herd immunity. Now let us understand the vertebrate immune system slowly, so that we can start forming the foundation of the immunology. Immune system has remarkable properties of recognition of foreign or non-self substances. That means any substance, any material, be it is a drug or a food or pollen or any chemical, which is not which does not belong to our body, and if it enters inside our body, then our body can easily recognize it. So our body can easily recognize, or our immune system can easily recognize the foreign or non-self substances. Then our immune system can also recognize what is self or our own body's substances or components. That means it can easily differentiate what is self and what is non-self. That is the remarkable property of the immune system. Okay. Then it can also recognize whether the self components are altered or they are unexposed substance or components, right? So when the new if they are self altered, like in case of the cancer or tumor, our body can easily recognize. Okay, so that is the ability of our immune system, that it can recognize self and non self components. Now, coming to the second part, it also has the remarkable property to respond. Now, the response is proportionate to the infectious threat to minimize collateral damage. Now, what is proportionate? Proportionate means that if there is a small threat, small risk, small infection, then our response is also small. It is not that we will give the high attack to a very small infection or very localized infection. Or in other words, if I make you understand, is that like if there is a local infection in a particular tissue, for example in a kidney, then not necessarily our whole body or the whole system will start producing or giving the response in each and every organ, because it is not needed. It is only needed in the kidney, it will be given in the kidney. So the response is proportionate. Proportionate means it is at the level of the infection. Lower level of infection, lower response, local response. If the infection is spreading, then we are our body will increase the response. And then to minimize collateral damage. Now what is collateral damage? Collateral damage is the condition where in order to fight the infection, our body produces certain chemicals, which we call them as a cytokines, or we call them different like interleukins, or we have different terminology which I will tell you. And they also give some sort of inflammation. So inflammation is the condition where our body, it shows the sign of redness, the sign of a bit heat, and then the redness and some pain. So when these symptoms come, we call them as a inflammation. And inflammation is a very very important aspect of immune system or immunity, or the kind of the fighting which is going on inside our body. It is a response against infection. But when this inflammation happens, it not only damages the microbe or the pathogen or any foreign agent, but also there are chances that the local tissue is also get damaged in that particular condition. Now you can understand something like that. In our society, we have a police and we have a army, right? And they have the role to protect us. In the same manner, our body also has the immune system which has the role to protect us. Now if there are some terrorists, right, and then terrorists, they have invaded a particular house, and then our army or our police, they have surrounded that particular house, and in the house we also have some civilians. Now what is the chances that when there will be the exchange of fire, right, there are chances that civilians might get hurt, either by the bullets of the terrorist or by the bullets of our own army or police. So this is known as collateral damage, right? The same thing happens in our body too. When our body is infected, right, and we have a pathogen, so in order to fight the pathogen, the aim is to kill that particular pathogen, to eliminate that particular pathogen. But in the whole process, there is a chance that, and it's a good chance, that our local tissues are also get injured. Now this is known as the collateral damage. I hope now you understand what the collateral damage is. What is the proportionate response, right? So these two terms are important: proportionate response and collateral damage. Then immune regulatory mechanisms or checkpoints ensure that this proportionality. That means our immune system knows where to stop. Our immune system knows that the organism or the pathogen is eliminated. Now it is the time to stop, right? So this is known as the regulatory mechanism which regulates the whole response. And once the response is over, once the pathogen is eliminated, then the all the cells, they become calm, right? So they, for example, like if everything goes good outside in the as I give the example of the terrorist, if terrorists are killed, then the army will return to its barrack, right? They will not be there. Okay, they know that the things the problem is solved, and now they will return to their barracks, or the police will return to the police station. In the same manner, our immune system, once the microbe or the pathogen is eliminated, then they will return to the normal state. Okay, and they will clean the debris and they will clean the microorganism from the site. The same thing happens in the society also. When there are some killings of such elements, then ambulance come and then these ambulances, they carry the bodies, they clean up the things, and some paperwork happens there, and then everything goes in the normal way. The same thing happens inside of our body too. When there is a microorganism, it is killed. When it is killed, then it's debris and it's the microorganism, it is removed. We have so many mechanisms, and then the things go normal. Okay, so our immune system has the remarkable properties of recognition and response. Now I will take you to some initial history of immunology. You know how this the fascinating science got foundation. Okay. Now, if you see the earliest written reference to immunity, it it was in uh, 430 BC. BC is before Christ. We call them before Christ. That's an old terminology. So that means today, if suppose we are in 2025, and first we will go to zero, the year zero, and then we will go back 430 years. So 2025 and 430. So it is almost 2455 years, right back. The earliest written reference to immunity by Thucydides. This Thucydides, he was the journal, okay, in the Greek army, and he described the Peloponnesian War of Athens. Now, if you see around 2500 years back, there used to be the city kingdoms, right? The city kingdoms means one city, and it was administered by one king. Then another city, it was administered by another king. So in that society, two cities, I think it was the Athens and Sparta, right? They had a fight because there were some other cities also, and there were two groups. The main leaders were the Athens and Sparta, and they fought for the dominance. And he was the journal, and at that time, the plague, it was the disease which actually spreaded like anything and killed so many people. So during the war, journal, he wrote what he observed, and his observation was very very important for the future research. What he observed, that those recovered from plague, right, could not contract second time, and that's a very important observation around 25 years 2500 years back. That means people who either they died, right, or they recovered, and when they recovered, when second time the plague hit, so they were protected. That was his observation. He did not know why it was so, but his observation was that okay, the people, they were recovered from the disease. So this was the earliest written record. Now why it is important? There might be some other observations also before Thucydides. There might be some other very important observations also, but we lost because we did not have, or we don't have the records. So this is the earliest record we have, and this record actually gave a very important information for the future refinement of the vaccination. Now, from this 430 BC, let us jump to the 15th AD, right? In the 15th century, we have the first recorded immunity by Chinese and Turks. Turks are the Turkish people. Okay. Now, at that time, smallpox was the disease, and this disease was very dreaded disease, and the mortality rate of smallpox was around 30%. So that means out of 100 people who had the disease, 30 would die. So that was the mortality rate. Okay. Now to prevent smallpox, they dried the crusts from the pustules. Pustules are the, if you have some understanding about the smallpox disease, then this disease will have the the pus filled small pustules. Okay. So what they did, they took out the pus and they dried the crust, and then these dried crusts were inhaled or applied onto the skin cuts, and this technique was called variolation. Why variolation? Because in French, la variole, that means smallpox, because they are fighting the smallpox, and they took this term variolation. Now in this picture what you can see, this gentleman, he is applying the dried crust into the nostril of a young boy, okay, using a small pipe. So he might have taken a small crust at this end and then directed the pipe towards the nostril and blew the air so that the crust can go directly inside the nostril. So this was known as the variolation. Okay, so this was the story from the Chinese and the Turks. Now let us move to India. In India, a tika or a small dot would be made on a child, usually on the sole of the foot by traditional tikadars. Now, in India, tikadars were the people who somehow gained this expertise, and what they used to do, they used to visit village after villages, and then in a particular season, smallpox is sometimes seasonal, you know they used to go during the outbreak or before the outbreaks, or maybe they may have defined some timelines, and then they used to apply the same thing, the crust, to the young ones by putting a small, in season, putting a small mark when the blood is oozing out, and then they used to put a paste with the oil and the crust applied on that particular wound. So this was known as a tika. Why? Because in the case of girl child, this incision was put at the forehead here, just like we put the tika, a small mark, very thin mark, and putting the paste and then rubbing it there, and then keeping the child in the isolation in a room. And then the ladies of the house, they used to make some rangolis kind of thing, on the floor, and then they used to do some pujas and worshipping the goddess, and the goddess particularly worshipped was Shitla Mata. Now Shitla Mata, it came from the Shital, the word came from Shital means cold, right? So because the smallpox disease, it had a high fever, high grade fever. So to in order to keep it cold, they used to started worshipping Shitla Mata. And the priest, because these tikadars, they used to be the priest in many cases, particularly in Bengal and Odisha, and they used to perform the some puja or some worshipping in the house for few days. And since they had applied the crust, smallpox crust, then the child usually get mild fever, and when it gets he or she gets a mild fever, then they used to think that it is the blessings of the goddess Shitla Mata which is coming to the child, and the child would be protected. Okay, so that's how it started, that the tika, the word tika is still used for the vaccines in Hindi. If you go, they will say the tika karan, or the tika is the term which is used for vaccines. So that is the origin of the term tika, and how the variolation was the very prevalent. It was very prevalent in our medieval Indian society. Okay, so in 1796, it was the Edward Jenner, you know, who inoculated a boy with a cowpox pustule fluid, later intentionally infected the child with the smallpox, because smallpox was the disease of the focus. you know a lot of mortality was happening up 30%. If you can see 30% in a population, it's a big number. We are not talking about the 100 200 people, but we are talking about maybe sometimes the thousands of people who are dying because of this disease. And it is not only the deaths, but also the disease is leaving a very ugly marks on the face and the body. So people were very worried about, these things, and nobody knew that the causative agent of the smallpox was a virus, because at that time we did not have this much of information. So they used to think, and in fact it was a global phenomenon, that the whole world used to think that it was happening because there is some, because the god was not pleased, and that's why, they created different folklores around the disease. So when in 1796 in England, this guy Edward Jenner, he inoculated a boy with a cowpox. So cowpox is a disease similar to smallpox in humans, but it was in cow or in bovines. So what he observed, now again the basis was the observation. What he observed, that the milkmaids, who were taking milk out of the cow, they were somehow immune from the smallpox. Okay. Now, because this observation and the observation, what was, we had the written record of the Thucydides. The observation, he made him think, sorry, he made, yeah, the observation was, like, it made him think to inoculate a boy with a cowpox pustule fluid, and then intentionally infected the child with a smallpox, and the child did not develop smallpox. So that was the first actual modern vaccination which was developed and applied by Edward Jenner. So that's why Edward Jenner is also known as the father of vaccination, right? Father of vaccination. Vaccination. Okay. So he pioneered the concept of vaccination by developing the smallpox vaccine, which is the world's first vaccine. And because of this success, the smallpox was successfully eradicated. Now there are no cases of smallpox virus on this earth in humans. The last known case of naturally acquired smallpox was in Somalia, which was recorded in 1977. So that was the last case. Now then came the Louis Pasteur in 1879, and he made the fowl cholera vaccine. Okay, this also has an interesting story where, he inoculated some chickens with the fowl cholera, which was a bacteria, and then the chickens died, and then he left his vaccine or his bacterial preparation and left for some vacation. And when he returned, and when he injected the fresh chickens with with that particular preparation which he left in the lab, he found that the chickens did not die, although they developed the disease, but they did not die. So what he conferred and what he observed or what he thought was that the the pathogen lost its virulence. That means its pathogenicity somehow, or reduced its pathogenicity, and that's why the fowl cholera, sorry, this yeah, the fowl cholera bacteria, it did not cause the death of the fresh chickens. So in 1879, this vaccine, it was developed by Louis Pasteur. Then in 1881, it was the anthrax vaccine for goats and sheep. And in 1885, the first vaccine which was tested in human was the rabies vaccine. Okay. And it was in a given to a boy named, I think, Joseph Meester, who was bitten by some rabid dogs. And since you know that rabies is a 100% fatal disease, so Louis Pasteur, he raised the vaccine and gave to this boy, and the boy recovered. And that was a huge success, and the news, spread it like a wildfire in the society, and everyone, it came to Louis Pasteur for the rabies treatment. And that was, the very, the advent of uh, or the, I would say, the it started the golden period of the immunology, where, we started working, and finally the foundation of the immunology laid down. Okay. Now two terms I will give you. After the success of the Louis Pasteur, now the people who were working in biology, they had a very clear cut observation about this, that something is working. Okay, it is the vaccines, they are working, but how they are working, nobody knew it. What was the reason? Nobody knew it. So later it was the Koch, Robert Koch, who came up with the idea of the germ theory, and he said that the germs are responsible, or the microbes are responsible for the disease. And so slowly the science grew. And when people started to evaluate the reasons of the immunity, the protection which was given by these vaccine preparations, then there were two separate groups, and the two separate groups found two separate things. Now one group, he found that it was some components which are present in the blood or serum, which were providing uh, immunity. So they called it as a humoral immunity. Why? Because humor means fluid. Okay, the term humor means fluid, and since this was, the soluble components were in the blood or serum, they called it as a humoral immunity. While the other group, it he the, sorry they came up with their own research, and their results were that there were some cells, you know and these cells were the phagocytes, and these phagocytes, they were engulfing the some bacteria or the pathogens, and so they called it that it is a immunity is cell mediated. So now two schools of thought, they cropped up. One was the humoral, and other was the cell mediated group. So now if we go further into the humoral and cellular immunity or cell mediated immunity, we will see that this humoral immunity, it is provided by different complements: mannose binding, lectin, C reactive proteins, lysozymes. They all are the part of the innate immunity. I will just tell you after a few slides what innate immunity is, but just understand that they are the part of the innate immunity, and antibodies, which are the part of the acquired immunity. So these all components are present in the body fluid, and therefore the immunity, because of these components, is called the humoral immunity. Now, during the search, some soluble components exhibited specific activity, and they were named accordingly. For example, some components neutralized toxins, so they were called antitoxins. Some components precipitated microbes or toxins, they were called precipitants. And some components, they clumped the bacteria, so they were called agglutinins. But later, all actions were credited to a single component, which is gamma globulin, and known as antibody or immunoglobulin, and they contained in the body humor or the body fluid. Okay, so that's how the humoral immunity is. Now antibodies are secreted by WBCs or white blood cells. These are the blood cells which are present in our body, and particularly a kind of WBC which is known as B lymphocytes or B cells. So in short, we can say B cells, otherwise the full name is B lymphocytes, and they confer passive immunity. Passive immunity is when the immunity is transferred from one individual to another. Okay, and this immunity is just a transferred immunity. We can call it as a transferred immunity. It is quick but short lived. That means it will immediately give the relief, it will immediately neutralize the pathogens, or it will immediately start working against a particular pathogen for which it is given, but it is very short lived. Once they are exhausted, it is over. For example, antivenoms, you must have heard that when there is a snake bite, Doctors give immediate antivenom. So antivenom are the antibody preparations which are given to immediately neutralize the snake toxins or scorpion toxins. Or maternal antibodies, they are also a kind of passive immunity. Passive means when the body is not making own antibodies, rather the antibodies are transferred, okay, and they are given passively. So in case of maternal antibodies, when the embryo is inside the body, then the mother's antibodies are also going inside the embryo and giving it protection, because the embryo is not making its own antibody and the mother's blood is circulated throughout the body. So maternal antibodies are an example of the passive immunity, and antivenoms are the examples of the passive immunity. Now if we see in contrast, we have the active immunity. And what is active immunity? It is the natural acquisition of immunity by microbial components. So when microbes, they come inside the body and our body fights, and then it generates antibody against those microbial components or the microbes, then we call it as a active immunity. This form of immunity is called as a active immunity, because our body is generating our own antibodies. Antibodies are not transferred from one individual to another, rather our body is generating the antibodies. So this form of immunity is called uh, active immunity. Okay. Now, cell mediated immunity, it refers to the protection provided by certain cells, right? As I told you that phagocytes, they are the cells, which were discovered. So T lymphocytes are a kind of WBCs. So we have just seen the B lymphocytes, okay? Here the B cells which are responsible to generate antibodies. Now we will see the T cells or T lymphocytes. They are again a type of WBCs. They are responsible for cell mediated immunity. And then we have the phagocytes. Phagocytes are the cells which are present inside the body, and it's a general term which engulfs the pathogen. Okay, engulfs the pathogen. So they are basically the microbe or non-self material eating cells. Okay, now there are few points to remember here. First, lymphocytes are mainly responsible for both cellular and humoral immunity. Now why? Because you see, cellular immunity is by the T cells and humoral immunity is by the B cells, and both are the lymphocytes. We have the T lymphocytes, we have the B lymphocytes, and they both are mainly responsible for cellular and humoral immunity. Then T cells confer cell mediated immunity and B cells confer humoral immunity, just discussed. Now T lymphocytes or T cells, they are derived from the thymus. Now you should know now why we call them as a T cell. Why this this T is coming here, because they are derived from the thymus. And B lymphocytes, they are known as B cells because they are derived from the bursa of Fabricius in birds, which is an outgrowth of the cloaca. So if you have studied the anatomy of birds, you will find that there is a small space, cloaca, near the tail, which opens up in the environment, and this cloaca receives it receives the excretory material as well as the sperms, so that they can go outside, so they can help in the excretion and the reproduction. So this is it's an outgrowth of the cloaca, and there this bursa of fabricius, they are formed in birds. And in the mammalians, for example we humans, we have bone marrow. So again it is B, bone marrow. In birds it is B, bursa of fabricius. So that's why these cells are known as the B lymphocytes or the B cells. Okay, right. Now both humoral and cell mediated system are intertwined. Now you should also remember that these humoral and cell mediated system, they are not separate systems, they are not individual systems, rather they are the intertwined systems, and both are necessary for a complete immune response against most pathogens. So they work in coordination. They never work individually. They work in coordination. And that's why if our body needs to give a complete immune reaction against a particular pathogen, then both the systems will be activated and they will be working together so that a complete response will be given to neutralize the pathogen or a particular agent. Okay, so these are the few points which you should remember, and that helps the formation of some foundation of the immunology and the concepts of the immunology. Now if we see the humoral versus the cellular immunity and how they together work, you can see this is the vertebrate body. Now here we have an example of mice. Okay, now we have foreign proteins or infectious agents. So here you can see three different kind of foreign proteins or infectious agents. So this mice is infected with these infectious agents. Okay, now the body is infected. When the body is infected, then what is happening here? We have the antigen. You can see here the antigen, right? Now this antigen, it is binding with the B cell receptor. We have the B cell receptor. These B cell receptors are actually the antibody molecules, the same antibody molecules which are secreted by the B cells, but they are membrane bound. Okay, they are not free form, but they are membrane bound. So this antigen, it binds with this B cell receptor, and when it binds with the B cell receptor, it gets activated, and when it gets activated, then the B cell starts secreting these antibodies. So here you can see the antibodies are secreting, and these antibodies are actually neutralizing the antigen. So the antigen is eliminated because these antibodies are part of the body fluid, and it is happening inside our body fluid or tissue fluid. Then we call it as a humoral response, and humoral response is given by B cells because B cells are generating the antibodies. Okay, now you come here. In the cell mediated response, we have the T lymphocytes. Now these are the T lymphocytes, and we have the T cell receptors. Now T cell receptors are not secretory as in case of the B cell receptors, which are the antibodies and which are secretory in nature. So T cell receptors, they bind with the antigen, and when they bind with the antigen, they form some sort of cytokine secretions. So our cytokines are the chemical messengers. Okay, in immune system, they are the chemical messengers, and these chemical messengers, they create a gradient and then they alert the neighboring cells. Okay, so they also go there, and they can also act upon the B cells, and they can also recruit the B cells at the site of the infection, and they also go to another kind of T cell which is known as the TC cells or the T killer cells, cytotoxic cells. Okay, so this is the T cytotoxic cells. Now you know that we have two kind of cells. One is the T cells, and this is also the T cell. This T cell is known as T helper cell. T helper cell. So this is the T helper cell, and this is the T cytotoxic cell. So these cytokines which are released from the T helper cells, they are not only going and asking B cells to come and to reach at the site of the infection, but also they are also stimulating the T cytotoxic cells, and these T cytotoxic cells, they are killing the infection. Okay, they are killing the infected cells. So they are the basically our snipers. They kill the infected cells. So that's how you can see here that we have the humoral response and we have the cell mediated response, and both responses are working together beautifully. You know, that's how they are not working in solo, rather they are working together. Okay, now we are coming to the concept of antigen. Now, antigen is any substance that elicits a specific immune response by B or T lymphocyte. Remember, any substance, whether it is a pathogen, whether it is a chemical, whether it is a drug, whether it is a pollen, whatever that is, if it can elicit or induce a specific immune response. Now, specificity is important here, which I will tell you. A specific immune response by either B cells or T cells, we call this particular substance as an antigen. Okay. Now, antigen plus lymphocytes. Now, lymphocytes, they can be B or they can be T, and that leads to clonal selection. Now, that's a new term, clonal selection. People who know clone, and I'm sure you know people who are watching this video, they must be knowing what a clone is. Clone is the identical copies of a particular organism or cell, right? Or a gene or a DNA, whatever that is, we call it as a clone, like a Xerox copy, right? So clonal selection is what? An individual B or T cell expresses a specific membrane receptor. When we say specific, that means it will bind to a single antigen. Where there may be millions of antigens, right? But this B or T cell, it will bind only a single antigen, or even not only a single antigen but a part of the antigen, which we call it as a epitope, and which in the further discussion I will uh, tell you what actually how it happens and what an epitope is. So this individual B or T cell, it will express a specific membrane receptors for a single distinct antigen. So you can see here the B cells, and you can see these B cell receptors, which are the antibodies, right? You can see the shape are like Y shaped, which are the characteristic shape of antibody. And then we have the T cells, and on the T cells we have the TCR or T cell receptor, right? Next, the specificity of receptor is predetermined before its exposure. Predetermined means the receptors which are present on the B cells or T cells, they will only bind to the very specific antigen, not all the antigen, and that is already determined where they are going to bind or which antigen they are going to bind. Antigen has not come yet, right? But they are ready with the receptors, and they know whenever the antigen will come, they will catch. So that's here, the this is known as the predetermined specificity, because specificity is already fixed, determined, and now we are just waiting for the infection to come. We are waiting for the right antigen to come, and then they will bind to it. So that's the second important thing. Then binding activates the cell. Now what happens when the antigen will come? It will bind, right, to this receptor, and it will activate the cell, causing proliferation onto a clone of cells. Now the situation is that there are so many cells in the body, and they are the B cells and the T cells. Now each B cell and T cell is different in terms of its specific receptor. So there are a good number of cells which have different receptors for the antigen, and when the right antigen comes and binds to the right cell, then this cell become activated, and once it become activated, it starts proliferating into a clone of cells. So now suppose it is just one or two cells in the body, and when the antigen comes and binds to these receptors, the cells will start proliferating and they will start increasing their number. So the same cell which was just one or two in the number, maybe now it is in thousands or in 10,000s, depending on the situation. So here you can see in the picture, Here you have different type of cells. The different colors are denoting the different receptor specificity. Okay, so this is the purple cell, so that means this is the purple specificity, this is orange specificity, this is green specificity, and this is blue specificity, for example. Now these are the two antigens, and these antigens, this is the star shaped and this is the cross shaped. Now you can see that these are two different antigens, and when these are two different antigens, they are binding to two different cells. Okay, so here this antigen is binding here with orange specificity, and this red one is binding with the green specificity, and they are not touching these two cells. Why? Because their receptors are not specific for these two antigens. Only the orange and green cells have the receptors which are specific to these antigens. Now after binding, the cells start proliferating. So initially the cell was just single, but now you can see after proliferation they have become four. One has become four. Okay, 1 to 2 and 2 to 4. So here green cells are also proliferating, so they have increased the number. So these are the clones, and finally they are secreting the antibodies. So this antigen is not only binding here, but depending on the number of antigens, they will be binding to the other cells also, and all the cells of the orange, they will start secreting the orange antibodies which will bind to the star antigen, and all the cells which are green, they will start secreting the green antibodies which will bind to the cross antigen. So that's how this is known as clonal selection. So based on the antigen interaction, a clone of cells was selected and a clone was formed after the proliferation, and that's how the response initiated and expanded. Okay. Now daughter cells have the same receptor specificity as the parent cell and engage more same antigens, as I just discussed. Okay, so now let us start with some important concepts of mammalian immunity. The first thing is that the pathogen must breach, I have written it in red. It must breach the physical barriers, and we have the physical barriers as a skin and mucous membrane. Now in order to enter our body, the pathogen should break the skin barrier and reach the tissue or blood. And this can happen only either when it breaches our skin or breaches the mucosa or the mucous membrane. So all the natural openings, they have the mucous membranes, right? Like our mouth has the mucous membrane, nose internal we have the mucous membrane. So if some pathogen is going inside the nose, and then this pathogen is breaching the mucous membrane, then it is going inside our body, it is easily accessing our tissue. The same thing happens if we have a skin cut, and the pathogen, environmental pathogen, can get enter inside our body. So in both the cases it must breach the physical barrier. Then the same pathogen may be treated differently in different parts of the body. Now, the pathogen is the same, but if it is going in different parts of the body, it is being treated differently. Now, this is how it happens inside the body. Okay. The immune response quickly fine tunes itself to counter the attack. So immediately, as soon as the attack happens, as soon as the invader goes inside our body, our immune system start recognizing, because you have seen that our immune system has the remarkable property of the recognition and response. So when the pathogen is recognized, and our body knows that where and which kind of pathogen is there, then it quickly fine tunes itself, it quickly optimizes its response so that it can give the best attack to the pathogen. The activated cells molecules in a given immune response depend on what kind of army of our cells will come, or what kind of army of molecules will come. It will depend on the chemical structure present on the pathogen. What kind of chemical structure is present, its presence, whether it is inside or outside of the host cells. Some pathogens can be extracellular, Some pathogens like viruses are intracellular, so their presence, whether they are inside or outside, if they are outside they will be engulfed by the phagocytes, right? If they are inside, then our cytotoxic killer cells will kill the host cell so that the pathogen will be killed. Then the location of the response, whether it is in the tissue or it is in the blood. If it is in the tissue, then we have a different set of cells and molecules. If it is in the blood, then we have the different set of cells and molecules. So it all depends, what is the location. So these things determine, the kind of immune response our body gives when there is any infection or any microbial invasion, right? Now, cytotoxic T lymphocytes, as I just, in the previous slide I discussed with you, cytotoxic T lymphocytes or TC cells, they detect and attack intracellular pathogens. That means those pathogens which are inside the cell. As I told you, the viruses, they are neutralized by the TC cells. They are specialists. They are the encounter specialist, right? So encounter specialist, what do? They kill the criminals. They have the skills. They don't ask anything. They know this is the criminal. They just shoot and kill the criminal. And that's how we call them as an encounter specialist. So our TC cells, they are the encounter specialist. They detect and they attack the intracellular pathogens. And B cells or antibodies, they recognize extracellular pathogens. So those pathogens, for example, bacteria or worms or fungus, which are in the tissue, right? They will be recognized by the B cells or antibodies. Okay, so our antibodies which are there, or our B cells which have the antibody receptors, they will find their suitable, the epitopes or the antigens, and then they will start proliferations, and then secreting the antibodies and then attacking it or neutralizing it. Then unique antigenic structures which are expressed on pathogens but not typically found in mammals. All the pathogens, because their origin is outside the body, they are not developed in, or they are not they originate in our body. So they have the different constituents on the surface, and this unique antigenic structures which are expressed on pathogens, okay, but not typically found in mammals, we call them as pathogen associated molecular patterns or PAMs. That means those patterns which are expressed by the pathogens, only on the pathogens but not by the mammals, which are very unique and which are the signature molecules. We call them as the pathogen associated molecular patterns. Remember this thing. So all the pathogens, they have some expression of molecules which are expressed solely on them but not on the mammalian systems or mammalian cells. They are very unique to them, and such receptors or such expressions or such molecules are known as pathogen associated. Because these are associated with pathogen, we call them pathogen associated. Then molecular patterns, because these are the patterns, they are not with a one time expression, but they are the regular patterns and they are molecular in nature, so we call them as a molecular patterns. So these are pathogen associated molecular patterns. Remember this term. Okay, then these PAMs are recognized by many cell surface and soluble proteins of invertebrates and vertebrates. So because these are the PAMs and our body has the ability to recognize them, so they are recognized by many cell surface and soluble proteins of invertebrates and vertebrates. Now cell surface proteins are like antibodies. On B cells we have the antibody receptors, and these antibody receptors can bind to the specific antigen. So this is the example of the cell surface proteins. And soluble proteins, again the antibodies which are present, or there are some complements or some other proteins like CRPs and some other proteins. They are the examples of the soluble proteins, and they are present in invertebrates and vertebrates. Because they are our own protein, they can easily recognize these PAMs, which are very unique structure. Okay, So WBCs express pattern recognition receptors. Now the pathogens express PAMs, but our WBCs, they express the pattern recognition receptors or PRRs. And these PRRs recognize polysaccharide coat of bacteria and some other common foreign structures. So they have, these receptors which can easily recognize the polysaccharide coats on bacteria and some other common foreign structures. So these PRRs which are expressed on the WBCs, these are conserved proteins. Okay, so during the course of evolution, these PRRs are conserved. They have not changed much. They are conserved proteins, and they are always expressed by many different immune cells, right? Because this is historically the interaction between microbes and the animals is, it's a very very long association or long interaction, millions of years back, when initially when there were only microbes, and then few small groups of animals, they started coming up, and then slowly the complexity and the diversity increased, so their interaction also increased. So some, because at initial stage, when they recognized or when they developed this interaction, the animal body developed the certain proteins which can easily recognize them, that did not change over the period of time, whether it was the part of the lower invertebrates or lower vertebrates or the human beings, they were conserved. So that's why it is written here that PRRs are conserved proteins, and they are always expressed by many different immune cells, and these are the first line of defense. Okay, they are the first line of defense for the quick detection of many common invaders. So as soon as such invaders or pathogens they enter into our body, immediately our PRRs, which are the conserved proteins, they recognize them. And once they recognize them, then the whole cascade of immune action, it starts. Okay. So these start a cascade to label and destroy the target pathogen, and these allow early profiling of the pathogen for an appropriate response. So once our body recognizes that, okay, this kind of pathogen is there, it is intracellular pathogen, it has such signature sequences, then the cells which recognize that, they come at the site and they start elimination of or appropriate response against the pathogen. So here you can see some cell associated receptors, like we have the TLRs, which are the toll like receptors, then NLRs, we have RLRs, we have CDS, CLRs, scavenger receptors, then we have N-formyl met Phi. These are the three amino acid receptors, and these are all cell associated. And then we have the soluble one are pentraxins, collectins, phycolins, complements. These are some of the examples which give immediate recognition to any pathogen which comes inside the body. So we will slowly, in our next discussion, we will go deep into the details of each and every, such receptor. Now, new and unknown antigens can be recognized. When any new and unknown antigen enters the body, it can be recognized due to the generation of diversity in unique recognition molecules by developing B cells and the T cells. As I told you, that the B cells and T cells, their uh, their receptors are already predetermined. When they come into the system, when they go into the blood and the lymphatic system, the receptors have the unique specificity, and it is already determined that which particular antigen they are going to catch. So this is generated, this diversity is generated during the development of the B cells and the T cells, and all the B and T cells, they originate from stem cells. So stem cells are the cells which give rise to any kind of cells. Okay, they are pluripotent cells, we call them as a pluripotent cells, and they give rise to a number of different progenitor cells, and further the progenitor cells give rise to the terminally differentiated cells. So here the stem cells, some stem cells are giving rise to, suppose, these four different type of B and T cells. But during the development, these cells, or some cells, for example number 1 and number 4, they start showing affinity towards our own molecules. That means they start recognizing our own body molecules as a foreign, and then start giving the immune response. Immediately they are deleted. So this is known as the deletion. So this is deleted and this is deleted, and this is happening in the primary lymphoid organs. So we have the lymphoid organs where the development takes place, and during the course of the development, when such cells, they come into the picture which can give immune response against our own antigens or our own body components, then such cells are deleted from the system, and only those which recognize self, and they know that this is self and we have not to act upon them, only those cells go into the circulation. So you can see here it is the circulation through the body. So cell number 2 and cell number 3, they are good to go and they are into the circulation. And when they will find a suitable antigen, for example an antigen has come whose specificity is with the cell number 2, the receptor is the antibody receptor which is here, it can recognize this particular antigen. So after binding with this antigen, it will give a number of clones, and you can see here a number of clones are generated. So that's how the clonal selection happens and clonal expansion happens. So once they select the clone, so here it is the clonal selection is happening where the specific antigen is binding with the specific B cell, and once this selection is happening, then expansion is happening, and expansion is simply the proliferation of the same type of cells to increase the number, so that more and more antibodies can be generated of the same specificity to deal with this particular antigen, because we never know, our body never knows that how many copies of such antigens are present. It may be present in millions, so we need more than a million antibody molecules inside the body. Okay, so that's how it happens. The process of self tolerance, as I told you, that the cells which are deleted from the system because they act upon our own body molecules, we call it as a deletion. But those which are sustained in the system, they are the self tolerance. They tolerate our own components, although they have the specificity, right? And maybe some may bind also, but they may not go beyond that. Some non-specific binding may happen but may not go beyond that. They may not start proliferating, they may not start attacking, but they are just tolerating. So this is known as the self tolerance. So the process of self tolerance ensures that the immune system avoids destroying host tissue by deleting self recognizing cells. So we have just discussed that the self recognizing cells are deleted. Okay, 1 and 4 deleted. Now immune system commonly ignores cancerous cells. Now this is actually the problem, but this is also the characteristic of a healthy system, a healthy immune system, that it also ignores the cancerous cells that arise in the body, as long as, now it is important, this is important, as long as these cells continue to express self structures that the immune system has been trained to ignore. So because our own cells are trained to ignore, okay, we know that these are our cells, so our immune system, it ignores the cancerous cells. Also they are dividing, their division, the division is going in a very fast way, but still our immune system does not touch them, because it knows that these are our own cells, and that's why the immune system is trained to ignore it. Now this is the problem also, because that in that case the cancer increases. But that is also the part of the healthy system, that our immune system is not attacking our own cells. If it start attacking those cancerous cells which have not yet started showing up the mutated proteins or the non-self proteins or the different proteins which our immune system is not trained to recognize, then our immune system can also attack some other organs or some other components. So that may create a problem, and that causes certain disease which is known as the autoimmune disease, which is also the part of the our discussion. Okay, so that is a separate thing, but as of now you should understand that our immune system is trained so that it will not recognize our own self molecules, but it will only recognize the non-self molecules, and this discrimination is the healthy characteristic of a immune system, right? Now, we had the original concept of tolerance as a self versus non-self. So tolerance is what? Our immune system is trained what is self and what is non-self. It can easily discriminate, right? That was the original concept of tolerance. Recently, there is a suggestion that danger or damage model by Polly Metzinger of NIH. So Polly Metzinger of National Institute of Health, USA, proposed a model, a new model or an alternative model about the danger or damage model, right? And that talks about the tolerance. Okay. Now, if you see in this picture, you will find that this is the normal cell. Okay. And this normal cell can be degraded either through apoptosis or through some mechanical or disease related failure. Now, when it is the apoptosis, then a number of components will be generated here which will not give any immune response. So only they will clean up. So our cells will come, phagocytes will come, and they will clean up the debris, but they will not induce any immune response. But in case of the necrosis or the necrotic cell, there are certain set of molecules which are released. These are known as alarmins. Okay, we call them as alarmins, and these alarmins are danger associated molecules. These are danger associated molecules, and these alarmins can activate immune cells. So that means our immune cells are activated by certain molecules which are released either by the pathogenic invasion or by some mechanical failure of the cell or the mechanical injury of the cell, and that set of molecules which can induce immune cells are alarmins, while such molecules are not released by the normal apoptotic cells, which is a part of our homeostasis, where the cells die and they are replaced by a healthy cells or a new cells. So here there is no immune response, but there is only a cleanup response. So this model by Polly Metzinger is known as danger or damage model. So immune system examines each new interaction for its potentially dangerous versus harmless to the host, then for whether it is self versus non-self. So that is a new recent idea about the concept of tolerance, whether our body really recognize the self versus non-self, and is that the basis of the immune response, or whether it is the type of the response or the interaction of the immune system with the potentially dangerous or the harmless, whether the interaction is a dangerous or harmless. So what is suggested here is that even if the pathogen is there and it is not producing enough dangerous signals, then it may be tolerated. Why? The earlier concept of self versus non-self was that any pathogen which enters into the body, if it is recognized as a non-self, whether it is giving, whether it is dangerous or not, but it is not tolerated or it will be eliminated. So this is here the new kind of concept or new kind of hypothesis about the tolerance, about the basis of the tolerance, an alternative view you can say. So sensing non-self without danger may not elicit an immune response. Sensing non-self without danger may not elicit an immune response. Now let us come to the discussion about the innate and adaptive immunity. Now these two terms, few slides back we used it, and now it is the time where we introduce these terms to you. So innate immunity is the form of immunity which is present from birth. Okay, so it is there in the body since our birth. It is natural or native immunity, and it is the primitive immunity. Okay, so the immunity which you can say, like, the God gives us as a gift, so that we can go in this world and we start fighting the pathogens from the day 0 or day 1. Then this kind of immunity is known as the innate immunity. While the adaptive immunity is acquired, it is gained, it is not present at the time of the birth, but during the development process, during the course of our life cycle, it is where we are exposed to different type of antigens, and then this immunity is developed. Okay, so it is dependent on the innate pathway. Again, this immunity is not something which is independent, rather it is dependent on innate pathways. So our innate immunity is the first line of defense, and then it gives the antigen to the adaptive branch of immunity, where it deals more vigorously, more aggressively, and that it eliminates. But this immunity is developed only during the course of development, and that is the reason why young kids, they get more infections during their childhood. If you see, most of the vaccinations, they are done in childhood. Okay, and particularly by the age of 2, so many vaccines are given to the children. Next, till the time they get adolescent, or they get young, they get frequent infections. And this frequent infections, this actually is good for them. Why? Because they are gaining immunity, and once their immunity is there, that will give them the lifelong protection when they become adult, and when they become at their old age also, till the old age also. So frequent infections are good. And why they are getting frequent infections? Because they have not got such infection earlier. So whenever a new antigen comes, they get infection. Whenever the new antigen comes, they get infection. But when this infection is coming second time or third time, then they are showing either the mild symptoms or they are not showing the symptoms. They are tolerating it because the body is fighting silently, and now they are not showing the symptoms. So this branch is known as the adaptive, as it is acquired. And the by birth, which the branch of immunity, and the all mechanisms which are present, it is known as the innate. So that is the basic difference between the innate and the adaptive immunity. Now second point is, immune response is non-specific. That means any kind of bacteria comes, virus comes, fungi comes, pathogen comes, or a worm comes, they have the same response. They don't discriminate whether this is worse, this is bad, this will be very ugly, whatever that is, they have the same set of response. Okay, you are coming, you are welcome, we will treat you with the same manner, no discrimination. So this is known as the non-specific response. While the adaptive immunity, it shows highly specific response. Okay, what kind of the pathogen is coming? If it is a virus, we have a different set of molecules and cells. If it is a bacteria, we have different set of molecules and cells. So such kind of specificity is seen in adaptive immunity, while non-specificity is seen in innate immunity. Innate immunity is quick response, immediate response within hours or days. They don't wait. You come, you will get treatment. But adaptive immunity, because it takes time to understand, the kind of response, and to tune up their own action, it gives some delayed response because they need time, okay, to develop that. So this is a delayed and this is quick response. That is another difference between innate and adaptive. Now, next is the response duration is short, you know because they don't have much things, they have the same set of things. So the pathogen comes and the response is immediate, but it is short because they are exhausted with the options, they don't have much options. So why they will take time? Whatever time is taken for the initial response, they take the time and then finish off. They will just transfer it to the adaptive, or the adaptive branch comes into the picture, right? But the duration is long. They keep fighting with the infection, and the duration is long as compared to the innate immunity. Next is that no memory of the antigen is formed. The antigen is coming, they are treating, antigen is eliminated, fine. Same antigen is coming twice, thrice, after frequently they are doing the same thing. They don't even recognize whether it has come again or not. They don't have that capability. But in case of the adaptive immunity, memory is formed. Once the particular antigen has entered and our adaptive immunity has treated it, then it also takes the picture of that particular criminal and keep it in our memory, that okay, when the next time it will come, we will recognize and we will treat it immediately and quickly. The same thing happens in our police station. The criminals come and a record is maintained, okay. As long as the crime is not coming in the light, the criminals keep doing crime. But once the criminal is caught and is brought to the police station, the police takes a picture, keep the complete record, all the marks on the body, this, that, and everything. Where do you live, the address, everything is recorded. And this record is there in the system. Even now we have the biometric also. If some crime happens and immediately there are some fingerprints of the particular criminal, and if it is matched with the database, then the response is quick, because now we know who the person is. We don't need to go and just start from the scratch, because we already have the record. The same thing happens here also in case of the adaptive immunity. Once the pathogen has been treated, if the same pathogen comes again inside the body, then we have the memory cells. Our memory is generated and preserved, and when the second time the exposure happens, then the treatment is very quick, and it's in a greater magnitude, okay. Then the repeat exposure has the same response. We have already discussed this. Again and again the same antigen will keep coming, it will get the same repeat response, the same response. There is no change because we don't have much. But here repeat exposure has heightened and more brutal response, right? It is just like if you have come first time, okay, we will treat you gently, but we will neutralize you. But second time, if you will come again in the body, the response will be very heightened and brutal, and it will be very quick. You will be immediately eliminated. So that's a hallmark of the adaptive immunity. Now these are some of the examples of the innate immunity. For example, skin, that is the physical barrier. It is the innate. Skin is present by birth. It is a barrier between the body's internal environment and body's external environment, outside the environment, external environment. So that is the barrier. Then we have the antimicrobial peptides. For example, we have beta defensins. That is one example. Then we have the complement systems. We have the gastric pH. In our stomach we have HCl, and HCl helps not only in digestion but also with food. If any microbe or pathogen comes in the stomach, then it is also destroyed. So we have the gastric pH which is very low, right? Then we have the TLRs, toll-like receptors. We have phagocytes. We have the natural killer cells. We have, the whole arsenals of molecules and cells which provide innate immunity. While the example of adaptive immunity is the T lymphocytes and the B lymphocytes. So B lymphocytes are responsible for antibodies. Okay, you must have now remember this thing. So they generate antibodies. And T, we have two kind of T cells. One is the T helper cells, and we have the T cytotoxic cells. T helper cells, they secrete the cytokines, and T cytotoxic cells, they are just our encounter specialist. They are the killer cells, they kill. Okay, they are the killer cells, and they are the part of the adaptive immunity. So this comparison will give you an idea about the difference between the innate and adaptive immunity. Remember this, because this is important, and these are the some foundations and the basics of the immunology which will help you in the future classes. Now, there are some cardinal features of adaptive immunity or the acquired immunity. What are these cardinal features? First is the memory. So, whenever a new antigen comes, it is treated side by side, its memory is also preserved. So, higher and faster response to subsequent exposure. So that next time, if it will come, we will have the higher response, we will have the faster response. Okay. Then we have the clonal expansion. What is clonal expansion? We have just seen. We increase the number of antigen specific lymphocytes. For example, first time we have just 2 or 3 lymphocytes in the body, for example. Okay. Now antigen comes and it binds to those 2 or 3 lymphocytes. When it binds, it starts a cascade which gives the proliferation, and the proliferation increases the number of the lymphocytes maybe to thousands. So from 2 to 3, now we have thousands of lymphocytes, and these thousands of lymphocytes, they have better chance to go and supervise and check or surveillance the tissues and cells and the other parts of the organs and body, because the numbers are more. Okay. Now that also gives an opportunity that this infection might be there in other places also. So the number which is increased here, it will take care of such infections. Then second thing is that because the now they have more proliferation and more number of cells, when the second time the infection will return, then we have a good number of cells already there along with the memory cells. Okay, because memory is already there, and we have different cells which are the memory cells, which we will discuss later. So in both the cases, clonal expansion and memories, they make sure that if the infection returns again, then it is treated very in aggressively and in a faster manner, right? Then the third feature is the specificity. All distinct antigens elicit specific response. We have antigens, and these antigens bind only to the specific lymphocytes, and they when they bind to the specific lymphocytes, they give the specific response, right? So this is the specificity. So we have seen this specificity earlier. Then non-reactivity to self. We have just seen the tolerance, so it prevents any injury to host tissues during responses against antigens, right? Then specialization. We have the tailored responses against different microbes. Tailored means we have different strategies for different microbes. If it is a virus, we have different cells and molecules. If it is a bacteria, we have different cells and molecules, and so on. So this tailored response against different microbes. And diversity. At last, diversity is what? That immune response against a large variety of antigens. Now this diversity ensures that whatever kind of antigen will come into our body, it will get treatment. That means our cells are capable to recognize them, although they don't know how many type of antigens are there outside. They are wandering here and there, and they don't know when they will come. But whatever kind of antigen will come, its part or the whole antigen, we have the cell to encounter that, and then it will be treated. It may take some time. Some infections are treated faster. Some infections take time because our lymphocytes take time to reach the site and to recognize the infection, but that's okay, it is a delayed, already we have discussed that. But definitely for the sure we have the cells which will make sure that any antigen which comes into the body, it will get treatment. In case if it is not get treated and it is inside our body, now there are two chances. Number one, it is harmless, okay, because our body's immune system is not working, our body will consider it as a harmless, and we will not have any symptoms and we will just keep doing our work. Or if it is not being treated and it is started giving its pathogenic impact, then definitely we are in trouble. And when we are in trouble, we need some assistance, and this assistance may be for bacteria in the form of antibiotics, or in viral case of viral maybe some antivirals, or some other medicines. So we need the assistance, so we have to keep these things in mind. Now, innate immune system can recognize about 10 raised to power 3 molecular patterns, okay, around 1000 molecular patterns of microbes, while adaptive immune system can recognize about 10 raised to power 7 or more distinct antigens. So you can see these numbers are very big, and since this number is very big, we can almost be sure that our body can take care of any infection or pathogen which can reach into our body. Okay, as long as we are healthy, we are taking nutritious diet, and we are keeping our body in good shape, our immune system will help us. In case if we will start doing something which is not good for our body or our immune system, then definitely we are in trouble, and then we will have to face the consequences. All right, so here is an example of innate and adaptive immunity collaboration. So how the innate and adaptive immune system collaborates. So you can see the infection, it can come here. It can come either through mouth, you can see here the throat infection, or you can see here in the digestive tract, you can see here, or you can see here through the cut or splinter, where you can get the infection because there is a breach. So we have the mucosal immunity here, and we have the infection agents which can breach here. So our mucosal immunity, if you can see here, the epithelial cell barrier is always there, and epithelial cell barrier, it keeps the infection away from the body, okay. But in case if the infection is able to, or the pathogen is able to breach this epithelial cell barrier, then we have some innate immunity come in action. Now phagocytes you can see here, and these phagocytes, they are actually taking up the infection, and then they are not only killing it, right, but also taking the sample to the lymphatic nodes or lymph nodes, right. Now these lymph nodes, they are present at some distances in the body, okay. They are just like you can see, or you can say them, these are the police stations, and these are the constables. So these constables, they are picking up this antigen and they are bringing them to lymph nodes, and lymph nodes through the lymphatic system. Now they have the filter, okay. These lymph nodes have the filter, and through this filter, these lymph nodes not only filter the lymph and circulate it to the further in the further circulation, but also they also have the opening of, or their the blood, where we have the B lymphocytes and T lymphocytes. You can see here the B lymphocytes, they are developed in the bone marrow, which is the primary lymphoid organ, and the thymus, which is the primary lymphoid organ for T cells, and bone marrow is the primary lymphoid organ for B cells. From the lymphoid organs, these B cells and T cells are generated, and then after maturation and passing the quality test, that is the tolerance, they come into the circulation, and in the circulation slowly they also reach at in the lymphocytes. So you can say then these are the police department, okay, police inspectors, you can consider them. So constables are bringing the the criminals, which are the pathogens here, they are not only killing them but bringing some samples, and the police is also coming here. And when they have the specific interaction, they proliferate. They proliferate, and they in here you can see the T helper cells, they are proliferating and then also secreting the cytokines, and these cytokines are acting upon the cytotoxic T cells, which are again proliferating. And here they are secreting the cytokines which are acting upon the B cells, and they are also proliferating, and they are giving rise to the antibodies. And all these cells, they again go into the circulation, and they reach at the site of the infection where more pathogens are there, and then they start killing. And that's how they control the infection. So first is the stage is the infection, then we have the innate immunity. Okay, then third, there is a transfer of the infection to the lymph nodes or the lymphatic system. And then we have the lymphocyte activation, differentiation, right? Then they are coming out, and then they are going at the site of the injury or infection, and they are giving the effector response. So both innate and adaptive immunity, they collaborating. So epithelial cell barrier, it is a kind of innate immunity, right? We have a cell barrier. Then we have these phagocytes, these are also kind of innate immunity. We have the molecules, these are the toxin molecules, they are also killing or controlling the infection. These are also the examples of the innate immunity. And here, if you see, we have acquired immunity in the form of the B cells and form of the T cells. So that's how and this is how our innate and adaptive immunity collaborates. Clear? Okay. Now moving forward to the primary and secondary response. I think we I have just discussed it but not given the terms. I will give you here. The first exposure with the antigen, it produces a primary response. The first response is known as the primary response, and it is because of the first exposure, when the first time the pathogen comes, enters into the body, and then gets treatment. This first exposure or the primary response is of low magnitude, short duration, and peaking at about 10 to 20 days. So here you can see this particular uh, graph, and here you can see this is shorter, this is low magnitude, this is the magnitude, so this magnitude is low, this is shorter. Okay, here you can see the time, and it is peaking somewhere around 14 days or 15 days, maybe 14, 15, 16 days. It is peaking. This is the peak, this is the peak here, right? So this is known as the first exposure. Now second exposure to the same antigen. Okay, the antigen remains the same. This is antigen A, this is the repeat antigen, and this repeat antigen is coming after some time. So here you can see by 28 days our immune response is 0. That means we have returned to our normal situation. Now this is the break. Now this break indicates whether it may be few weeks, it may be few months, it may be few years. But when this antigen comes again, now you can see this is known as the secondary response. And this secondary response is greater in magnitude. So now you can see the peak here. Okay, you can see this is the magnitude, this is the first magnitude here, and now you can see the second magnitude, the second exposure magnitude is here. So this is the greater magnitude. Okay, then it peaks in less time. Now you can see here it takes around 0 to 14 or 15 days to peak, but if you see here it is peaking in around 7 to 10 days. Okay, so here you can see, and sometimes in some cases it can peak in 1 to 4 days also. So the peak is less, sorry, in less time, and it is more antigen specific, right? Because it is already we have the memory here, so it is more antigen specific. Now we our body can easily recognize the particular antigen, and it can again start giving a very quick response, faster response, and a higher response. This first exposure and second exposure is the foundation for vaccination. Now you know that vaccines are given. So when vaccines are given to an individual, and if it is not exposed naturally, then the first response comes. And when first response comes, and then after some time when booster dose is given, so booster dose, maybe the first booster dose or second booster dose, it gives the subsequent second exposure, secondary exposure. And secondary exposure is basically increasing the immune response, increasing that particular memory cells inside the body, so that if in future naturally the same antigen comes, then our body is well prepared to attack that in a very higher and faster way. Okay. Now there are some unusual immune responses, and these unusual responses are because of some immune dysfunction or failure. When there are some failure or immune dysfunction, then we get some unusual immune responses. First is the hypersensitivity. Hypersensitivity, like including allergy, it is over attacking on common antigens. You remember that we discussed that the response should be proportionate. When the antigen is giving a small impact, our response should be proportionate only to control that particular thing. But here, if you can see, it is over attacking on common antigens. Antigens are very common. For example, pollens, for example, dusts, they are always every time everywhere, but our body is reacting in a very aggressive manner, which it should not, and that gives to the situation which we call it as a hypersensitivity. So this is a kind of immune dysfunction. Okay. Then we have autoimmune disease. Autoimmune diseases are the diseases where our own immune cell targets self protein. Okay. So that means when there is a breakdown of self tolerance, then our immune system starts attacking our own body, and that causes autoimmune disease. Now this is the example. You can see some gout like situation, okay, or some arthritis. You can see the joints are swollen, and the antibodies are formed against our own joints. And then it causes inflammation. Okay. So that is one example here, autoimmune disease. Then we have immune deficiency. Immune deficiency is the insufficient immune response due to absence of any critical immune component. So whenever, either in case of innate immunity or in adaptive immunity, if some or other components are missing, then it causes immune deficiency, and our immune system is not able to protect us. Okay. And then we have another example, which is the immune imbalance. Now here we have an example like chronic inflammation. Chronic inflammation is that the body is not having any infection, but still we are having inflammation. Inflammation is a response, right? And this inflammation is chronic, long term inflammation. Although body is it is always constantly at the state of in the state of inflammation, and it is causing the immune cells to work, and that is producing disease. And that is nowadays it has come up through the research that many diseases like cancer or cardiovascular diseases, they are because of the chronic inflammation. Even dementia is because of the chronic inflammation. So it is very important to understand that it is not only the activation of the immune system or the deactivation of immune system can cause immune imbalance, but also the inhibitory pathways where the immune system is brought down to the normal situation. If that breaks down, that also creates a problem, and that causes the immune imbalance. Okay. So it is the disturbance in immune inhibition post activation. After activation, when it is the time to inhibit the response and to calm down the response, it doesn't happen there. In transplanted tissues and cancer, the healthy immune system rejects or destroys non-self proteins, such as those encountered in most transplant situations, and tolerates cancerous cells. So now we have two situations. One is the transplanted tissues, and then we have the cancer, which is the self altered cells. So our healthy immune system, if the immune system is healthy, it will reject transplantation. So transplantation is rejected. That is the characteristic of the healthy immune system, right? So that's why, doctors prescribe immunosuppressants, which suppresses the immune system so that the transplanted organ or graft is retained in the body and it is not rejected. Or destroys non-self proteins, and they also tolerate cancerous cells. So as we have just discussed that, the cancerous cells are tolerated because they are the self cells, and they are the self cells, so our immune system is trained to ignore such self cells. Whether they are fast dividing, it doesn't know, that it may be the problem. It only knows that, they are our own cells, and I will not have to kill them. So it is tolerated. So these two situations are the examples of the healthy immune system. Okay. So now we are just, closing our discussion, and what we learned today is about the immune system. We learned about some history. We learned about the humoral versus cellular immunity, then some important concepts of mammalian immunity. We also understood the difference between the innate and adaptive immunity. We also understood what is primary and secondary response and how they are important in vaccination, and then some examples of unusual immune responses. Okay. So I hope, by today's discussion you have gained some understanding about the immunology, about the immunity, and how the whole immune system works to protect. So this was a basic discussion, a foundation about immunology, and in the subsequent lectures we will go into the details. By the time, you should and you can, repeat this video again to understand few concepts, and if you have some feedback to give, you can always reach out to me. Thank you very much, and take care of yourself.